Hello, I'm Dr. Stephen Pipe from the University of Michigan, and on behalf of my co-authors, I'm pleased to be presenting first data from the Phase 3 HOPE-B gene therapy trial. This is an efficacy and safety trial evaluating Etranacogene Dezaparvovec. This is an AV5 capsid vector that includes a hyperactive factor IX transgene, the so-called Padua variant. And it's being evaluated in adults with severe or moderate severe hemophilia B. And what's notable from this trial is these patients have been treated irrespective of pre-existing anti-capsid neutralizing antibodies. We had previous insight on this particular AV5 vector from a study of AMT-060, which was a phase 1/2 trial. That trial used this same vector, but it had a native form of factor IX. And those patients now have been followed out as long as 5 years, and they've shown stable expression of a wild-type factor IX out through that follow-up period with no late emergent safety signals. A phase 2B study showed a mean factor VIII activity of 2 years at 44.2% that included this Padua variant. So that was the impetus for then exploring this vector with the hyperactive factor IX in the HOPE-B trial. And this is the first phase 3 study in hemophilia B to report, and it also has the largest gene therapy cohort to report results to date. The subjects who participated were a mean age of 41 years, but had a range from as young as 19 years all the way up to age 75. The vast majority of these had severe hemophilia B with less than 1% factor IX activity. Notably, based on the insights we had from the previous study, there was no correlation of neutralizing antibodies with efficiency of transduction. So even though all these subjects had neutralizing antibodies measured, they were not excluded from the study. And it turns out that 42.6% of the patients had neutralizing antibodies. The factor IX activity has now been followed out as long as 26 weeks for the entire cohort, and the mean factor IX activity at week 26 was 37.2%. We do have individuals who've now been followed even beyond that point, with the longest follow-up period being 18 months after dosing, and there does not appear to be any evidence of a loss of expression over time. Importantly, of the 42% of the individuals who had a positive neutralizing antibodies, there was no correlation with the pre-existing neutralizing antibodies and the factor IX activity they achieved. There was one subject, however, who had a titer of 3,212, and this subject did not respond to the treatment. As far as the efficacy, once patients achieved a steady state, it was really impressive reduction in their bleed rates. Total bleeds were decreased by 83%, and treated bleeds were decreased by 91%. And if we look at the entire cohort, almost all of the patients, 98% of the patients, have been able to discontinue prophylaxis. There were 2 subjects, the one patient who did not respond, and then there was one other patient who had a transfusion reaction, and unfortunately he only got 10% of the dose, and so he didn't get a full dose and did not have a response to transduction. With regards to overall safety, the majority of the treatment-related adverse events were mild. 9 patients did have transaminase elevations, and they received a steroid treatment per protocol, and all were able to discontinue their steroid use prior to week 26, and the factor IX activity was preserved in the mild range between 8 and 39% in those subjects. So overall, first report of a phase 3 study in patients with hemophilia B, the largest gene therapy trial cohort to report to date. Mean factor IX activity significantly increased to near normal levels at 26 weeks. No requirement for prophylactic immunosuppression, and 52 of the 53 subjects receiving a full dose had a response to treatment. Bleeding was abolished in the majority of patients through the follow-up period, and the safety profile looks consistent with the early phase studies. So the final analysis is planned for a 1-year follow-up, and that will give us the durability of the factor IX expression, and then also the ability to characterize the bleed control over the full 52 weeks. And so based on evaluation of that data, decisions can be made about possibly submitting for regulatory approval.